All right, welcome back. Today we're digging a bit deeper into statistical genetics.
Yeah, building on what we talked about last time with molecular markers, those DNA signposts.
Exactly. Now we want to see how scientists actually use those signposts to find the genes behind important plant traits.
And for this, we're drawing heavily from, well, actual course materials.
That's right. We've got transcripts and class notes from Professor Guilherme da Silva Pereira's statistical genetics course. He teaches over at the Department of Agronomy at the Federal University of Viçosa.
So yeah, a real academic perspective on this.
Our goal today, to really get a handle on mapping populations and this really fundamental concept called linkage disequilibrium.
Or LD, for short. It's key to the whole mapping process.
Okay, so let's start there. Linkage disequilibrium, LD. What exactly is it in simple terms?
Okay, basically it means that alleles at different spots, different loci on the DNA, aren't associating randomly.
Meaning they show up together more or less often than just pure chance would predict.
Exactly. Think like allele A at one spot and allele B at another nearby. If they were totally independent, the chance of getting A and B together would just be the frequency of A times the frequency of B.
Okay.
But with LD, that AD combination might pop up way more often than expected, or maybe less often. It's non-random.
Right. But why does that happen? Is it always because the genes are physically close?
Ah, well, that's the crucial part. The LD we want for mapping is caused by genetic linkage genes or markers being physically close on the same chromosome. They tend to get inherited as a block.
Because recombination doesn't happen between them very often if they're close neighbors.
Precisely. But, and this is big, but LD can also be caused by other things.
Like what?
Things like population structure, distinct subgroups within a larger population, or maybe selection favoring certain combinations. Even just random genetic drift over time can create these non-random associations.
So associations that look like linkage but aren't actually due to physical closeness on the chromosome, that sounds like it could cause problems. False leads.
It definitely can. You might think two things are linked, when the association is really just because of shared ancestry or something else entirely. This creates false positives in mapping studies if you're not careful.
Okay, so if you have all these other factors potentially muddying the waters, how do scientists isolate the LD? They actually care about the kind caused by real genetic linkage.
That's exactly where mapping populations come into play. This is the solution.
Ah, okay. So instead of just sampling a wild natural population.
Right. You create a specific population through controlled crosses. You carefully select the parent plants.
And why the controlled crosses? What does that achieve?
It's all about minimizing those other confounding factors. By starting with known parents, offer ones that are quite different genetically.
And different for the traits you're studying.
Ideally, yes. Contrasting parents are good. When you make a specific cross, like an F1 generation, and then maybe an F2 or a backcross, you create a situation where most of the non-random association you see between markers and traits is due to linkage. The population structure stuff is largely removed because you designed the population structure yourself.
Makes sense. You're controlling the background noise.
Exactly. And the choice of parents is vital. You need that genetic diversity to actually see segregation, see different combinations of alleles appearing in the offspring.
And does the way the plant normally reproduces matter? Like self-pollinating versus cross-pollinating?
Oh, absolutely. It
Wow.
affects how LD breaks down over generations. In sulfurs, heterozygosity decreases rapidly, which changes how you set up and analyze the mapping population compared to outcrossers where heterozygosity is maintained. The starting point, like an F1 from two inbred lines, is genetically uniform, but it segregates dramatically in the F2. That segregation pattern is what you use.
So this leads to different strategies, like linkage mapping versus association mapping.
Right. Linkage mapping typically uses these purpose-built populations, F2s, backcrosses, RILs. You directly measure recombination frequency between markers of the trait by looking at the offspring. You calculate things like LOD scores to see how strong the evidence for linkage is.
And association mapping, or GWAS?
That usually uses natural populations or collections of diverse lines. It leverages historical recombination that's accumulated over many, many generations.
Which gives potentially finer mapping resolution.
Potentially, yes. But because you're dealing with all that complex history and potential population structure,
You absolutely need those statistical controls you mentioned to account for ancestry and relatedness.
you got it. Otherwise, you get swamped with false positives. Remember that hair color, skin color example? Often associated due to ancestry, not necessarily tight linkage. GWAS needs to correct for that.
And you mentioned population size being important too.
Definitely. You need enough individuals to capture a decent number of recombination events. The source notes mention needing at least, say, 100 to 250 individuals for a decent map in many crops. More individuals generally means better resolution. You can pinpoint the location more accurately.
Okay, so pulling it together, mapping populations are like these specially designed genetic experiments.
Pretty much.
Yeah.
Created through controlled crosses to create a clean genetic background where the main reason markers and traits stick together is actual physical linkage on the chromosome.
Allowing scientists to connect those DNA signposts to the genes influencing the traits they care about.
Exactly. It's a powerful tool.
Okay, fascinating stuff. So here's something to think about. How different do you think the overall genetic landscape, the patterns of LD across the whole genome might look in a species that primarily self-pollinates versus one that mostly outcrosses? And how would that fundamental difference in reproductive strategy change how you'd even begin to approach finding the genes for a specific trait in those different kinds of species? Something to ponder.